Okay, so let me do some clicking here real fast and show you those new clicks. So we're going to go down here, and if you remember, here's the end of exam 2. So exam 3 starts, we've done pheromones, cohorts, and there were 2 clicks until this weekend on environmental hormones. But there's some really good stuff. Some of it will go pretty fast for you, but I'm going to go through each click and tell you things to concentrate on and things to skip over. Because sometimes it gets detailed. They'll name a specific compound, like let's say an insecticide, and they'll say maybe it has androgenic properties. I'm never going to ask you that specific stuff because you can always look up a compound and see what it acts like. So we're looking at general things. Here's the video on environmental hormones. It's a good discussion, so think of it as a lecture that we would have done dead week, but we're going to do it now. And then Wednesday's photo period, and there's that line drawn right there. Okay? So let's go through this. And then you know when you click on these links, whatever shows up you're responsible for, but don't do more clicking on that page to go other places. There's one little exception to that, and that's like images. And I'll show you how to enlarge one. So here we go. Endocrine disrupting chemicals. Now you know from your physiology that mammals, and let's say horses or dogs or cats or whatever, make hormones. And there's usually one or more tissue that makes a specific hormone. It gets released into the blood, goes throughout the whole body, and only has effect on what's called the target tissues. And it's a very good principle. The target tissue is, in my analogy, if the key is a hormone, it's the only door that this key fits into and then does some action. Okay? Now here's a new little concept that we haven't maybe had before. The hormone is called the first messenger. And we're going to use some examples of steroid hormones. So let's say testosterone is released. By the way, everybody in the room right now is making testosterone. Everybody. So that's the key. It goes around the body, and it only has effect on tissues where the key fits in and it turns. It's like, think of it as starting a motor. But what it does inside the cell usually increases some other molecule, and that's called the second messenger in this series. The hormone is the first messenger, but then something is usually increased inside the cell, and that's called the second messenger. Okay? So these endocrine disruptors mess up our normal system. And the lecture is very good. It's a woman that's doing some studies, and they talk about how some cosmetics have compounds that act like hormones and all this kind of stuff. So I wanted to review a little bit in general the hormone stuff, and there are these 3 A words that you should know. A hormone agonist, a hormone antagonist, and a hormone analog, right? So agonist, that's A-G-O-N-I-S-T, agonist. A hormone agonist is any compound that mimics, and this is some of the words that I use on this website, mimics or acts like the parent compound. So let's say we have a progesterone agonist. If you inject that into an animal, it will have similar actions to progesterone. Okay? That means, and here's the kicker, that means it somehow interacts with the receptor. You can't have action unless the agonist interacts with the receptor. Okay? So it has to bind and do some action, maybe not totally exactly like the native compound, but it's got to interact with the receptor. That's this whole thing about hormones and specific receptors. So then a hormone antagonist is a compound that works against or the opposite of the parent compound. So if you have progesterone, which is a very famous hormone, and you say, I have a progesterone antagonist, if that's injected into an animal, it will work against progesterone's action. And what's the world's most famous, powerful progesterone antagonist? RU, that's 2 capital letters, hyphen 486. RU 486. Do you know that will work in any animal that relies on progesterone? It's not species specific. Because remember, steroids are common across all species. Progesterone looks the same in this room, everybody's making some progesterone, as it does in Africa on the female lines. So that's an interesting thing about steroids. It's similar all the way around. So we've done agonist, we've done antagonist, and then we have analog. And I have looked up the definition of analog for you. Here's how it's usually spelled right here. Here's my spelling of analog. A-N-A-L-O-G. A chemical compound that is structurally similar to another. And that's all we're going to say about that. It's an analog. It looks like it, but we don't know what it does unless you do experiments. And that's what all these chemical companies, all over the world, they make a compound and then they do some tests. And whenever you do a test with a chemical, like a hormone, on a biological system to look at some endpoint, you call that a bioassay. Okay? And some of this reading talks about they transplanted a rooster's testis into a female, the hen, a female chicken. And then they see what that's doing. That's called a bioassay. Now when you take a testis and put it in another animal, look how many things could result in the recipient. So it's not hormone specific, right? Because there's a lot of hormones that are made by the testis. We always think of testosterone, but there's others. So you got to be careful. The best way is to do a bioassay is, let's say, and I've done this in classes, let's say I have a compound that I want to see if it has any androgen activity. You can take baby chicks, and let's say somebody gave me this compound. Oh yeah, I got a good story about a barber. Okay. Anyway, I've done this in classes. You take these chicks, and you take this solution, and you put it on their comb. Every chick has no matter what their sex is, that red comb on top of their head. And so you have like a control, and usually steroids are dissolved in some kind of oil, like peanut oil or sesame oil, because they don't dissolve in water. So if you're ever doing experiments, it's got to be something that's got some oil in it. And lo and behold, if this unknown compound has androgenic activity, the combs on the little chicks will grow when they normally wouldn't. Okay? And that's called a bioassay. So you can have different compounds and see what happens. So here's what happened to this one barber. This is all hormone stuff that's not in the reading, right? The reading you can do. I'm telling you things that you don't know. This barber, and years ago, you don't know this, but years ago there were barber shops where only men went, right? And they had their hair cut by old guys, and there were no women around. Now you go into these beauty shops and you get your hair cut and you're sitting next to somebody else that's getting a haircut or their perm. Anyway, this barber had a lot of older gentlemen as customers, and they were balding, losing their hair. We won't point to anybody in the room that that's happened to. But he had this ointment that he would rub in his hands, And then every customer, that wanted it would get the scalp treatment. And after about half a year, now this was in the New England Journal of Medicine, so I'm not making this up. After about half a year, his breasts started growing. The barber, the male, he's a primate. Primate breast in the male will respond to estrogen. So he's doing this treatment on everybody, but he noticed all the customers, how often you get a haircut? 6 weeks, 8 weeks, right? So any one customer wasn't getting a lot of the ointment, but he was. So then, he goes to his physician, my breasts are growing. And then you know that there's some hormonal influence. So actually the first thing you would look for is a tumor that's making estrogen. So they did a lot of tests. They couldn't find any tumor. Finally, the physician had him bring in the lotion, and they analyzed it, and the lotion had a high concentration of estrogenic activity. It may not have been estrogen, but it was like an estrogen agonist. And so through transdermal administration, he was getting a lot of estrogen, and the target tissue was his breasts, mammary tissue. Very interesting. Okay, so that's analog. I think, so it's a chemical compound that's a structure similar, but then you don't know the action. That's usually the classic definition. Okay, so then I'm going to go back to this thing here to show you. So so when you're reading this, I would just read it once. I would have some notes beside me and try to pull out the biology and the general principles. So on this first one, there's not really a whole lot. Look, we've already talked about DES, right? That's a synthetic drug. There's the terms, right? Phytoestrogen, that's any compound that comes from a plant that has estrogenic activity. So then, this is kind of more of an introduction, okay? And then there's the old steroid molecule. So that one's that. Pretty straightforward. The next 4 links, are a review of hormones. Sorry, get my coffee here going. So this one is talking about androgens. So what are androgens? That's the family name of a bunch of compounds. So it's like the Johnsons. So you never really inject androgen into an animal. You inject testosterone, which is in the family of androgens. Okay, be aware of that. All these steroid hormones are synthesized in the same way in every tissue that's making them. So now some of the figures are clickable, and I wanted to show you that on this map here, this diagram. That's too small to read, right? But you can click on it and enlarge it. And I want to point out a couple things. Here, all steroid synthesis starts with cholesterol. Okay? Then, the next molecule is pregnenolone. So that's something to know. Cholesterol goes to pregnenolone, and then you've got a series of other reactions. Every step is an enzyme, a specific enzyme. So how would you prevent pregnenolone from being synthesized? You would have an enzyme inhibitor. Whatever enzyme is up here that catalyzes that reaction. So anytime you want to stop a steroid from being made, you give an enzyme inhibitor. And I'll give you one example here shortly. So I want to go down here to testosterone. This is testosterone, and look at estradiol. That's a female hormone, well-known female hormone, so-called female hormone, and dihydrotestosterone. So testosterone is the precursor molecule for estradiol synthesis. And that happens in the gonads of both sexes. Both gonads make testosterone, and in the female, there's usually more estradiol produced than in the male, but that's not always the case with every species. And that enzyme is aromatase. So I looked up aromatase. Here it is. So I'm giving you some specific enzymes, not all of them, but just a few. So aromatase, the enzyme involved in the production of estrogen that catalyzes testosterone to estradiol. Okay? So if you wanted to stop estradiol production, you would find an aromatase inhibitor. Okay, back to our diagram. Then, testosterone could also be shuttled to dihydrotestosterone. And dihydrotestosterone is an androgen. And it's more powerful than testosterone. And the prostate likes dihydrotestosterone. So in the prostate, that's one of the male accessory sex glands that enlarges in old men and old dogs, right? When the prostate sees dihydrotestosterone, it undergoes hypertrophy. And you can think to spell hypertrophy, but pronounce it hypertrophy. And in some people and dogs, it's called benign prostatic hypertrophy, BPH. They advertise these things on TV. Okay? So then, what happens is in the prostate, testosterone comes into the prostate, it converts testosterone to dihydrotestosterone by this enzyme. So now I'm giving you some enzyme names. 5-alpha reductase. So 5-alpha reductase takes testosterone and makes dihydrotestosterone. And that's going to happen in the prostate. Okay, so drug companies go, okay, the human population has trouble with benign prostatic hypertrophy, and we know it's because T goes to DHT. DHT is the abbreviation for dihydrotestosterone. So if we gave men an enzyme inhibitor that stops 5-alpha reductase, we could theoretically reduce the size of the prostate. And lo and behold, that's one of the treatments for benign prostatic hypertrophy. I think I told you Jennifer, the girl that was my graduate student that liked human stuff, and we did the alcohol, the fetal alcohol syndrome. She worked with this too. We did with this in boars. She talked some drug company into giving us the enzyme inhibitor. At that time it was called Proscar. And we gave it to boars to see if we could reduce the amount of dihydrotestosterone. So that's the enzyme that does this reaction. So aromatase goes this way, 5-alpha reductase goes this way, and it's all a simple conversion. Okay, so be aware of that. Now some of these, and this is like a review, it's talking about hormone docking. Okay, so here's the quick synopsis of that. If it's a protein hormone, it usually binds to the receptor on the cell membrane. And it's facing outside, so it's on the cell membrane. Steroid hormones usually have an intracellular receptor. So steroid hormones are relatively small, and they go through the cell membrane and bind to a receptor inside. Okay? That's a general rule. And some of these pictures have some clickableness to them, but sometimes the computers don't have the right programs. It doesn't really matter. I won't click on them and do anything with that. Anyway, so it's talking about androgens, androgen disruptors. Then these are compounds that are like blocking androgens effects. So you're saying the disruptors are really androgen antagonists. right? Okay, but they'll talk about specific compounds like this one here. I'll never name a compound and say, what actions does it have? Estrogenic, androgenic, whatever. I'm never going to get that specific. Okay, so then these other cliques are corticoids, and everybody knows where corticoids are synthesized from. The adrenal gland, that's half the right answer. The adrenal cortex. Okay? So here's another thing, a little sidebar. All the hormones that come from the adrenal cortex are steroids. All the hormones that come from the adrenal cortex are steroid. Okay, so then it goes through basically just like for the androgens. I think this is another clickable thing, but it's the same darn diagram. It has to be. See, that's a chart of what's called steroidogenesis. That can never be different. It's always got to be that way. Testosterone can be made to estrogen. Testosterone can be made to DHT, dihydrotestosterone. So that can never change. So anyway, they go through corticoids. Most of them are called glucocorticoids. Okay? And then they name some. Here's maybe the most famous corticoid, cortisol. A common stress hormone, right? But there are other corticoids. Anyway, you can read that, some little history, and then estrogens. We have that chart again that can never change. They talk about effects. So once you read one of the hormone things, the others are based on it. So there's progesterone, okay? Effects. So here they've got the ester cycle of some animal. It might be humans, not sure. But the point is, they show you the fluctuations of hormones. And that's one other thing. In some of the reading, and here's something you got to be careful about hormones. Somebody will say, oh, we did this study and progesterone levels were higher in group B than group A. You really got to understand hormone stuff to see if the data is true. What if they goofed up? What do you call that when the data is goofed up? Confounded. Be careful, because sometimes hormones can't be measured accurately with a one-time sample. Sometimes it takes many samples to get a pattern of secretion. Sometimes you can stress an animal and mess up its hormones. So what I would really like to do is, I know this is not practical, but somebody says, oh, this group of cows had higher estrogen levels. I want to see how they did the blood samples. Were they rough? Were they was it constant all the way around? Or did they change the time of day when they made the hormone measurements? It's scary out there how many times the data is confounded. And sometimes it's not obvious. So then they go through we're on progesterone, okay? Progesterone disruptors. See, they're going to name compounds, but I'm never going to ask you all these compounds. You just should know that there are some compounds out there, the wild yam, that mimic hormones. Mimic means agonist. Whenever you see mimic, that means agonist. And so hopefully when you read this stuff, you understand that it can mimic it a little bit. Like it can be a weak progesterone, or it can be a medium progesterone, or it can be a stronger progesterone than the native compound, right? The whole gamut. Okay, so that's enough of that hormone stuff. Then I guess there's a review of thyroid hormones too. So you read that, and you'll get the point here that thyroid hormones are basically T4 and T3, right? And T4 is basically not very active. It has to be converted to T3, and that's really the more active hormone. And so it goes through the construction of thyroid hormones. And then the actions. This is where we get really into the disrupting. So it gets to be hormone binding. Because you have to have hormone binding or binding of a compound for it to work, for some action. So you go through here, hormone receptors. You should know the number of receptors in a tissue can vary. If a tissue that's a normal target tissue doesn't have any receptors for some reason, then it doesn't respond to the hormone that it normally would. There's an R word that describes the tissue that doesn't respond like it normally would. Anybody know? You've got this tissue, and let's say you inject some hormone into an animal, and usually something reacts. Let's say, like I've done a lot of injecting estradiol into overactimized heifers, and they show heat, just like a normal heifer would. I can fool anybody. But what if I inject estrogen and the heifer doesn't come into heat? I would say she is, quote, unquote, with this word. Anybody know? Because you got to know, sometimes things don't work like you think. Refractory. R-E-F-R-A-C-T-O-R-Y. I think the spellers out there, right? Refractory. If a tissue is refractory, it doesn't respond like the textbook says it would. Because a lot of textbooks won't tell you, hey, there are cases where what we just told you doesn't work. Okay? And sometimes one reason for refractiveness is the tissue didn't get primed with another hormone. Sometimes one hormone goes to a tissue and it increases the receptors for another hormone. Okay? And one good example would be oxytocin. Everybody knows if you inject oxytocin, you get uterine contractions. That's one of the classics. But it basically has to be an estrogen-sensitized uterus. So sometimes you can inject oxytocin into an animal and get no uterine contractions because there aren't receptors for oxytocin because it wasn't exposed to estrogen beforehand. Okay? So it gets complicated. So this action stuff is very good. That one concentrates on receptors, transport proteins. When you read this, be aware that most hormones in the blood are bound to a transport protein. So in the blood, let's say testosterone. You can find testosterone 2 places in the blood. Free testosterone, which means if you look at it with a microscope, but you can't see testosterone, but if you could, it's just in the plasma by itself. And free hormones are able to bind to receptors. So free hormones are the active component. But then you have testosterone bound to proteins. And those are called androgen binding proteins. ABP, androgen binding proteins. And if testosterone is bound to androgen binding protein, it's not biologically active. But it's reserved. It's a reserve. Okay? So you need both components because once the free is used up, some of the bound becomes free. And that's what they're talking about here. Transport proteins. Okay? Is it bound or is it free? That's basically the two possibilities. Okay, so here, some hormone transport proteins in plasma are highly selective, maybe only transporting a steroid or a thyroid hormone. And here they call it sex hormone binding globulin. I said androgen binding protein. Sometimes you'll see them named globulin, sometimes protein. But be aware of this, that they have to be free to be biologically active. Okay, then we're on to metabolic changes, okay? And again, they're going to name some specific compounds, but I'm never going to ask you about those. But they go through, and that's a relatively short subject here, short writing. But look at some foods can affect a balance of hormones, okay? And result in goiter. So cabbage, too much cabbage can actually cause overproduction, oversize of the thyroid gland. Anyway, so that's something to think about. I wouldn't ask you exactly what does cabbage do, but you should know in general that some foods can affect the size of the thyroid. And we all know what goiter means, enlarged thyroid. Then, we're getting here about natural hormone production values, okay? And that's the other thing. They vary with age. And that's the other thing you got to be aware of when somebody reports hormone levels. Was the treatment group all the same age and all the same physiological state? Or was it a grab bag of animals and your value doesn't really reflect true values in some of the animals? Liver function affects steroid levels. If your liver is not functioning well, you tend to have longer half-lives of steroid hormones. And they'll be higher. And in some cases, some animals might be so metabolically active that hormone levels are lower in those animals because of their metabolism. And there's the transport protein levels change, okay? Okay, natural hormones are more potent than any of the known synthetic, okay, interlopers, okay, so DES, whatever, okay. Of course, dose matters. Okay, so here's a good word. These are 3 good words right there. Okay, additive effects. If 2 hormones have an additive effect, here's what you do. Write down 1 plus 1 equals 2. That means hormone 1 and hormone 2 added together to bring out a bigger result than either one. 1 plus 1 equals 2. Okay? Then synergistic. What would you write down? 1 plus 1 is greater than 2. That's a synergistic relationship with hormones. 1 plus 1 is greater than 2. And then if hormones are antagonistic on their effects on an animal, you would write 1 plus 1 is less than 2. Okay? And that's additive, synergistic, and antagonistic. So sometimes the presence of another hormone might accentuate the results. Now what we're talking about, like remember the chick and the comb weight? You could, when you do that on chicks, you could actually cut off the comb and weigh them for a bioassay. And if you had 2 hormones that you gave to the chicks and the results were basically similar to the 2 effects of the hormones separately, then you would say that's an additive effect. Or you could have a synergistic effect. 2 hormones bring a result bigger than the 2 independently occurred. Okay? Okay, I'm done. That's it. Wednesday, photo period. Have fun. Or not. The choice is yours, I guess, right?